We are delighted to bring you this seminar presented by Labroots. Welcome everyone, and thank you for joining us for today's keynote presentation titled, The Massively Parallel Sequencing Revolution. It is my pleasure to introduce you to Dr. David Smith, Professor and Consultant, Department of Laboratory Medicine and Pathology, Mayo Clinic. Dr. David Smith received his Ph.D. in Biochemistry from the University of Wisconsin in Madison in 1978, studying antibiotic resistance in bacteria. His laboratory studies the common fragile sites, which are regions of profound genomic instability that are found in all individuals. Dr. Smith is also the chairman of Technology Assessment Group for the Mayo Clinic Center for Individualized Medicine. For a complete biography on our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will respond back to you via email. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Smith. I will now turn the presentation over to him.
Hi, I'm David Smith. I'm a professor and consultant in the Department of Laboratory Medicine and Pathology at the Mayo Clinic. I am also the chairman of the Technology Assessment Committee of the Mayo Clinic Center for Individualized Medicine. Today, I'm going to be talking about the massively parallel sequencing revolution. An abbreviation for that is MPS. All of the things that I'm going to be talking about came about as a result of the technology revolution. And all of the sequencing technologies are highly dependent upon technological advances that first enabled one to generate the necessary data, but as important is the ability to analyze the data that is generated. All of the ability to analyze the data is predicated upon advances that have occurred in computers. With the doubling of computing speed every 18 months, all one has to do is compare today's laptop computer to the best computers that were available, the supercomputers 10 years ago. That's how much computing technologies have moved forward. And the key features in the advances in computers are the computing speed, or the cycles, having sufficient RAM and data storage, because for when we talk about things like sequencing complex genomes, we're talking about massive amounts of data that needs to be taken and analyzed. Everything that I'm going to talk about today involves the analysis of DNA. And when we think about DNA, we have basically 4 nucleotides, A, C, G, and T. However, there is an additional nucleotide that has to be considered, and that is 5-methylcytosine, which is the methylation of cytosine, which is an epigenetic modification. RNA can also be analyzed, but the technologies that I'm going to be talking about in my talk, which are first-generation sequencing technologies, you first have to convert that RNA into cDNA to copy it with something like reverse transcriptase. What I show here is the actual structure of a double-stranded DNA molecule that is in the process of replicating. As the 2 strands come apart, what happens is you have the capability of making a copy of each strand. The information on any one of the strands is sufficient to get the copy of the second strand. So as that DNA replicates and as it unfolds, if you have a T, the only base that can base pair with that is an A. If you have a G, the only base that can is C, etc. Now, this is the actual structure of just a linear DNA molecule. And what we have at the top is just sort of a caricature of that, showing sort of the backbone, and then the nucleotides themselves are the things that are in the middle. At the bottom is actually a structure of what it actually looks like with the atoms themselves. This is the canonical double helix that we all talk about. However, the DNA that we have in humans is not existing as just this naked molecule. On the next slide, we see that the DNA double helix, which is shown at the top, which is directly equivalent to what I showed on the previous slide, is actually wrapped into chromatin. There are actual proteins that bind to that DNA and begin to do a wrapping process. The first step is you have sort of beads on a string, which is a very loose form of chromatin. That can be compressed down into a chromatin fiber, which are actually packed nucleosomes. On the second slide, on the second thing, you see actually the nucleosomes, which are just the wrapped DNA around the protein. That chromatin fiber is further compressed to be an extended section of a chromosome. That can be even further compressed to be a condensed section of a chromosome. And when it's fully compressed, one has the metaphase chromosome, which is the way that chromosomes were traditionally visualized. This slide actually shows the morphologies of each of the human chromosomes after they have been banded with something which is called G-banding. And G-banding is the use of a stain called GIMSA. With GIMSA staining, you can actually, there's actually two pictures here. One is basically just an electron micrograph showing each of the chromosomes, and then the small little insert next to it shows the actual banding with G-banding of the individual chromosomes. And you have the largest chromosome is chromosome 1, all the way down to the last autosome, which is chromosome 22, and then the sex chromosomes, the X and the Y chromosome. If one prepares a metaphase preparation of these chromosomes, this is what they look like. And you see sort of just looks like little spaghettis of the individual chromosomes. While it's relatively easy to discern larger chromosomes from smaller chromosomes, it's very difficult to tell the difference between chromosomes that are of the same size. However, with the G banding, with the GIMSA staining of the chromosomes, you suddenly have a banding that enables you to take this mix of chromosomes and actually assort them and align them to say which are the two chromosome 1s, which are the two chromosome 2s. And the picture shown here is actually a metaphase, a karyotype of a human female. You can tell because there are two X chromosomes on the bottom right. You see the two chromosome 1s, the two chromosome 2s, and with the banding, it thus becomes easy to discern one chromosome from another. Obviously, the larger chromosomes are easy to sort of look at than, for example, the small chromosomes, chromosome 21 and 22. Now, this karyotyping and the ability to look at chromosomes in this way was very powerful, because with this it became possible to look at different diseases and abnormal states to see if they had anything to do with alterations that occurred in the chromosomes. And one of the very interesting early findings is shown on this next slide. This is the karyotype of an individual who has a chronic myelogenous leukemia, otherwise known as CML. The chromosomes look pretty normal chromosome 1, normal chromosome 2, etc., as you start to go through the karyotype. But all of a sudden, when one looks at chromosome 9, you can see that there are two different looking chromosome 9s. Similarly, when you look at chromosome 22, you also see two different chromosomes. What has occurred in this individual, what has occurred in every leukemic cell in this patient with CML is actually a balanced reciprocal translocation. A little piece of chromosome 9 has come off of that chromosome and has translocated to chromosome 22. The other piece has moved to chromosome 9. And this is shown on the right. You basically have this, the normal chromosome 9 versus the 9q plus. This is the chromosome that has a tiny little piece of chromosome 22 attached to it. Similarly, with the chromosome 22, you have the normal chromosome 22, but the 22q minus where the tiny piece has moved. Now, what has occurred in this? And this look and this karyotype revealed something very interesting at the molecular level, because what is actually occurring at the position where these chromosomes have broken off is that two genes have been translocated to each other. This is what is called the Philadelphia chromosome. This was called that because the investigator who found this was working in the city of Philadelphia. And basically, this is a balanced reciprocal translocation. And as a result of this, two genes, one called BCR and the other called ABL, have actually been translocated next to each other. And this occurs in 90% of the patients who have CML. Now, ABL is actually a tyrosine kinase. And when BCR and ABL have been translocated next to each other, and again, this occurs in 90% of the patients with CML, what happens is there is a promoter on the BCR region which activates ABL. And the activation of that tyrosine kinase is the trigger that triggers the development of CML. Well, it turns out imatinib is actually a tyrosine kinase inhibitor. And investigators, once they realized what was occurring in these, came to the realization that if they use a tyrosine kinase inhibitor, this might inhibit the growth of the CML cells. And indeed, this proved to be true. And as a result of this, this completely changed the course of chronic myelogenous leukemia, because with the addition of imatinib to patients who have this translocation, again, in 90% of the patients with CML, the survival rates, which were less than 30% prior to this advent, increased over 70% as a result of using this tyrosine kinase inhibitor to inhibit the activated ABL, which actually occurs in a majority of the CML patients. Now, this thus raises the question, are there thus easy fixes for other types of cancers? It turns out there are other translocations that have been found in both leukemias and in solid tumors, but none are as frequent as the BCR-ABL translocation in CML. Now, when one compares alterations that occur in the leukemias to the alterations in solid tumors, traditionally there are less rearrangements in leukemias than in solid tumors. So one of the next questions still remains, because of this real understanding that finding through cytogenetics, by looking at the chromosomes, that these two genes would juxtapose, one wonders, can we do similar things for other cancers? And would this give rise to understanding that might suggest therapeutic things that could target those cancers that have specific alterations? Well, here is a normal female. You can see again the karyotype, and the two X chromosomes aren't on the bottom right, they're in the middle row to the right, but you see the two normal X chromosomes. This female was seen at the Mayo Clinic because she had a mass inside of her abdomen, and when this mass was looked at, they realized that there was something cancerous that was growing in that individual. When the cells from that mass were taken and looked at, this is shown, the karyotype that is now shown on the bottom is the karyotype of what's something that obviously looked like an adenocarcinoma, which is compatible with an ovarian adenocarcinoma. And what you can see immediately, as compared to the very simple karyotype, which was seen in the individual who had the Philadelphia chromosome in chronic myelogenous leukemia, you see basically a genome which is dramatically rearranged. There isn't just two chromosome ones, there's actually three that are slightly different from each other. There's only a single visible chromosome 2. There's 4 different things that are all compatible with some chromosome 3s. And as one goes through the entire karyotype, you can see that almost every single chromosome, with the notable exception of just a few, have some alterations. And at the very bottom, there are a series of chromosomes that it's difficult to tell exactly what they are at all. They're more than likely representative of lots of different chromosomes, pieces that have come together. Thus, when one looks at many solid tumors, ovarian cancer is a more rearranged solid tumor, but many solid tumors have much more complex karyotypes, have many more rearrangements. And unlike the CML, where you have something that occurs in the majority of patients, it seems like every single solid tumor has a completely different karyotype, and there haven't been any great insights to find something that is occurring in many of those cancers that could then be a therapeutic target. So how many alterations occur in cancer? Well, the real only way to know is it's not sufficient to use cytogenetics and karyotype analysis. The only way to know for sure would be to develop the technological capabilities to determine the complete nucleotide sequence and structure of a large number of cancer genomes as compared to their matched normal genomes. In much the same way that on the previous slides I showed you the karyotype of the female with the ovarian cancer and then the karyotype of the cancer, well, what we would like to do is not karyotypes because they don't have the resolution. We would like to compare the nucleotide sequence of the normal DNA from that individual to the nucleotide sequence of the cancer DNA to see what are the alterations. How hard can that be? Well, it turns out we inherit 3 billion base pairs that are present in 23 chromosomes from our mother, and 3 billion, although men have slightly less DNA, we're a little genetically deficient, from our fathers. Unfortunately, what makes it extremely difficult, aside from the actual number of nucleotides, we're talking about 6 billion base pairs, is that 45% of our genome is highly repetitive. So in order to determine the sequence of a genome, let alone a cancer genome, you first have to figure out ways to determine the sequence of pieces of DNA. And then once you've figured a way to actually determine the sequence of the tiny pieces or larger pieces, you have to figure out a way to string it all together. So the very first technique for DNA sequencing was developed by two scientists, Maxim and Gilbert, and they had a chemical cleavage method, and their names ended up being on that method, for determining DNA sequence. Unfortunately, this was a very cumbersome technique and very low throughput. In spite of that, Gregor Sutcliffe in 1978 was able to use this technology and to develop the sequence of a plasmid called PBR322. This is about a little over 2,000 base pair plasmid, and using this technique, he was able to do this. This became his Ph.D. thesis. Unfortunately, this is a very cumbersome technique. It costs $10 to $20 or more per nucleotide. This is not going to be a feasible method for sequencing things that are much larger. However, a number of years later, a technique was developed by Fred Sanger that was, we call it Sanger sequencing, but its full name is di-deoxy chain termination sequencing. The way this works is to use chain terminating nucleotides in the synthesis so that you can determine where the certain bases are. If you have a dideoxy A, this will terminate when that is added to a growing chain. And the very first generation of this technology was done by having the actual nucleotides, the dideoxy nucleotides, being radioactive. So when they were incorporated, you had radioactivity that was incorporated into that growing chain. What I show here is actually how DNA replicates. This is reminiscent of a much earlier slide where I showed the two chains of DNA coming apart, but now we're looking at how a base is added to that chain. You have a template and a primer, and this is actually how sequencing is done, but it's also how DNA actually replicates in vivo. So when you have the primer, which terminates at this base pair between A and T, the next available base is a G. The only base that can base pair with this is a cytosine. So if cytosine is added, you will have a C added to that. But there's a hydroxyl group, which is the group that can now accept the next nucleotide. The next base is an A, so the only base that can come to that is a T. And you can see that we've now added a G and a T. The next base that would be added would be another T. Sanger really realizing this said, you know what if we were to add di-deoxy nucleotides? Di-deoxy nucleotides will not have hydroxyl group at the right end of the base that's being added. They'll just have a hydrogen. So when they're added, only a single base can be added. You cannot add a second base. So utilizing that knowledge, you see here actually how di-deoxy sequencing is done. You have 4 separate reactions, a reaction which is interrogating the additions and the positions of every single A in a chain, one for G, one for C, and one for T. The way Sanger sequencing works, however, is to have a very dilute concentration of di-deoxy nucleotides so that on the average, most of the time, a regular base is added. But when a di-deoxy base is added, that terminates the chain. So if you do these reactions, you can see in the di-deoxy A lane that there are 3 different bands that occur. Those are bands because they're bands where di-deoxy A was added either at the first position where there was a second position, or the third position, a T, and you can then do the same thing for the G, the C, and the T. And then by looking at these things, you can actually read up, and this is shown on the bottom, you have an A, a G, 2 Cs, etc., and you read up to get the sequence of that little stretch of DNA. All of the di-deoxy reactions that are done, and they're done separately, have to be resolved on large gels. And this shows a typical plate assembly where you have a plate that could be 2 meters in length, 1 to 2 meters, and it's very thin. You will load the DNA reactions, the dideoxy reactions at the top of this thing. You will place this into an electrophoresis apparatus with a positive electrode at the bottom. The DNA is negatively charged, so it'll run towards the bottom. And when the dye has hit the bottom, you know that all the bases from short bases to much longer bases will be on that gel. So after the gel has been run, you can peel apart the two plates, put a piece of autoradiographic film against it, expose it overnight, and then what you get is a series of bands. And the actual sequence can be read up by looking at it. You have a lane for A, a T, a G, and a C. Now, the problem with Sanger sequencing, or most of the sequencing we'll be talking about, is that you are sequencing short stretches of DNA. Fortunately, with Sanger sequencing, it is actually possible to resolve things that are 800 to 900 base pairs in length, but we're talking about genomes, which are considerably larger. Well, one of the early realizations was that a very easy way to use these short sequences to characterize longer sequences was to generate a set of overlapping fragments and sequence them and then align them next to each other to look for overlaps. And in this way, and what I show is just an average gene, which is 5,000 to 10,000 base pairs in length, can be characterized by these shorter pieces of DNA to build up a long sequence. And actually, one of the interesting things is Craig Venter was the first person to say if that's possible to look at a gene this way, it should equally be possible to perhaps take an entire complex genome and do it in the same way. Now, unfortunately, using di-deoxy sequencing and radioactive nucleotides, how many sequencing lanes with this type of technology would it take to sequence the entire human genome? Well, it turns out if you can do 700 to 900 base pairs of sequencing on a gel, but you require 4 lanes to determine the sequence of one fragment, and you have a 6 billion base pair genome, it would turn out to take 222 million lanes, which is 20 to 30 million gels. And this would take 1,000 graduate students working 10 years to do this. This is not, and the cost at this point was less than Maxim Gilbert sequencing, but was still a couple of dollars per base. Now, the next step was to actually figure out a way to automate this, to make this more efficient. And actually, once they did this, it thus became possible in the year 2000 to use Sanger sequencing. And this was predicated on first subcloning the little pieces into bacteria, because for Sanger sequencing, it requires at least 10 to the 13 molecules per every sequence lane. And you can do this by either sequencing with directed sequencing, knowing which fragment you have, or as Craig Venter suggested, completely random as shotgun type sequencing. But what needed to be developed, as shown on the next slide, is basically to instead of using radioactive dideoxy nucleotides, to actually use fluorescent nucleotides. And you can use 4 different colors, one for A, one for C, one for G, and one for T. And once that was done, and this was developed by Mike Hunkepeller, was to basically be able to put all 4 reactions into a single lane, run this on a gel, and then look at the bottom of the gel as the fluorescence came off, and each time you would see a color come off, you could say, oh, that was a color indicative of an A. And this is the sequence of about 300 base pairs of a fragment. Now, this still had to be further improved to be efficient enough for whole genome sequencing. So the revolutions were that the first generations of this were still done on gels, but once they switched to capillaries, reusable capillaries, you could put many samples through the same capillary on successive runs. And while the first generation had a single capillary, then they realized that you could do multiple capillaries, for example, a 6 capillary sequencing device, all the way up to a 96 capillary device. When this became possible, we had the development of a machine around 1995 that was known as the ABI 3700. This was a 96 capillary sequencing machine, which is shown on the slide. Basically, you have 96 simultaneous reactions, which you can put on a plate. Those reactions will then run through these 96 capillaries. They will run, and then a CCD camera at the end can determine the sequence. With this automated sequencing machine, it had the capability of generating 75,000 base pairs of sequence each time the machine ran, and the machine would run in 3 hours. With this type of thing, and this is the vision of Craig Ventner, here is a picture from Celera Genomics in Gaithersburg, Maryland, of the room that contains 330 of these sequencing machines. This is the sequencing factory that thus has the capabilities of tackling something as complex as Craig Ventner's genome, and his genome is the first named genome that was sequenced. So with 96 capillary sequencing and a 3-hour runtime, they were able to generate 31 million base pairs of sequence a day, and thus in 3 to 4 months could generate the complete genome sequence of Craig Ventner. And when they put it all together, they didn't just use the sequencer and sequencing by di-deoxy sequencing. They also used the efforts of physical and genetic mapping to help to string this all together. They also did not map the entire genome. They only mapped about 85% of the genome because regions that were highly repetitive, it was very difficult to determine when you sequence them exactly where they came from. Now, once this was done, the greatest impact was that it provided information on genome organization, which accelerated all biological research. These are high throughput methodologies, rapidly developing, but the actual cost, the sequence cost for doing this on these ABI 3700s was about $200 million. So what we need is dramatic improvements in this, 1,000 million-fold improvements in sequence output at decreased costs, and this led to the development of what are now called next-generation sequencers. The cost, again, they say it's about $3 billion. That's to buy the machines and get everything set up. Well, the reason that it was so expensive, as I mentioned before, is every single piece had to be individually cloned into E. coli, and each individual reaction was 10 microliters, but you had to do millions of these tiny fragments for an entire genome. The two together make it so expensive. So to bring genome sequencing to the masses, you have to figure out to dramatically decrease the cost of genome sequencing. It raises the question, how much would a genome have to cost before it becomes a routine part of standard clinical practice? But first, you have to solve the two main problems, the cloning problem and the cost for the reagents. Well, all of these are called massively parallel sequencing. As I said before, this is MPS, and they're all MPS technologies. So first thing you needed to do was to find a way to amplify individual molecules separate from each other. Unfortunately, because of this, it limits the fragment sizes that you can do to about 200 to 1,000 base pairs in size. Well, to figure a way to amplify them, the first thing is to replace cloning. And there were two different PCR-based strategies. One is called emulsion PCR. The second is called bridge amplification that were used to amplify fragments. But those two are both dependent on PCR amplification. The third is a PCR-free strategy to amplify, and that's called DNA nanoballs. Now, clonal amplification is shown here. In emulsion PCR, basically you make a water and oil emulsion, which puts tiny little water droplets that only hold about 50 picoliters in an oil emulsion. And what you do is you have tiny little beads. The beads have nucleotides on them, oligonucleotides that are attached to them. The DNA fragments that you are trying to sequence, or the entire genome, is fragmented into pieces. The pieces have oligonucleotides attached to the ends of them, which are exactly complementary to the nucleotides, that oligonucleotides that are on the bead. So if you have a tiny little water fragment and a tiny little bead with a single piece of DNA, that can be a PCR generator that can amplify that individual fragment. A separate droplet that's away from this one will have another piece of DNA, and it will amplify that piece of DNA. And the separate droplets enable you to amplify individual fragments of DNA to very large numbers. The second strategy, which is not dependent on these oil emulsions, is bridge amplification. Here, the oligonucleotides, which on the previous one were on the surface of a bead, are now on the surface of a flow cell. The DNA that you want to sequence is broken into pieces. Oligonucleotides, which are complementary to the oligonucleotides on the flow cell, are put on the ends, and you have a DNA, the fragments are flowed across the slide surface, and then one end will have an oligonucleotide complementary to many of the oligos on the thing. If that attaches, the other end can bend over and find its complement, and now you have a bridge. And the bridge can then enable you to start with a single fragment to make 2 copies of the fragment, and when those are denatured, you can repeat this process, and what you end up with is clusters that are amplified sequence of the individual fragment. And on the surface of the flow cell, you can have lots of individual clusters without the need for the messy oil emulsions. However, the first 2 techniques are still PCR dependent. Now, the third technique is to use DNA nanoballs. This was something that was developed by Rade Dramanjic and co-workers at Complete Genomics. What this does is you clone the individual pieces of DNA into a little adenoviral vector, and then you use a 5' polymerase, which basically runs into rolling circles to copy the DNA without the PCR amplification. This is a linear amplification. Each individual molecule is copied to at least 300 copies. Those tiny little piece of linear DNA folds up into something that's called a DNA nanoball, and those nanoballs can then be deposited into a pattern flow cell, which is shown on the right of that slide. So you now have the 3 different techniques for amplifying DNA. The second thing is how to reduce the reaction volume. Again, the 10 microliter reactions use a considerable amount of reagents. So there's 2 different techniques that we use to decrease the reaction volume. The first strategy was to put clusters on a slide surface. The second was to have small beads on a slide surface or in with wells. So the clusters on a slide surface are done basically with the bridge amplification. The small beads on a slide surface or within wells are what is done basically with emulsion PCR. I didn't describe it here. The third strategy, of course, is the DNA nanoballs, which can then be deposited into the little wells. All of this then is what leads to massively parallel sequencing. You're using technologies analogous to massively parallel computing, where in computing you just have thousands or hundreds of thousands of chips, each doing a small bit of the computation. Well, in massively parallel sequencing, each individual sequence is a small bit of sequence, but if you do this on hundreds of thousands or millions, you're doing a large amount of sequence. The very first sequencing machine to develop used picotiter plates. Picotiter plates were made by basically using fiber optic cables, which have 1.5 million fibers in them. You slice the cables like a salami, coat the bottom of the plate with plastic, and you have these tiny little plates that contain 1.5 million wells. And the very first commercially available massively parallel sequence was from a company called 454, and it was called the GS20 for the Genome Sequencer 20 because it could do 20 megabases of sequencing. Remember that the ABI 3700 did 75 kilobases of sequence per run, so this is a dramatic improvement. The process overview is to first amplify the individual molecules in emulsion PCR. So you ligate the primers to the DNA, you amplify them on the beads, you purify the beads that have amplified DNA, and then you put the beads with amplified DNA into the picotiter plate. That plate, with the necessary reagents for base incorporation, can be placed into a machine, and then you can flow first the base A, and to see if any of them have incorporated an A. The 2 enzymes that are also present there are the enzymes for pyrosequencing. They convert a base incorporation to a flash of light. So you have a camera at the top that watches when you've flowed A through it to see if any of the wells have incorporated an A. Then you flow the second bit, T, C, and G to see, and you can repeat this process. As a result of this, and you can see this on the right, you can actually determine the sequence of that individual bead. But remember, this is a picotiter plate with 1.5 million wells, so there are at least 500 to 600,000 wells that have an amplified bead on it, and that's how many sequences you're determining simultaneously. Now, the strengths were that the reads were long. It started at about 200 bases, but very soon they got to 400 bases. Then they made some improvements to make it even longer. But the weakness was that it was emulsion PCR, which is a cumbersome technique. The second one is that actually the bases that are being incorporated are unblocked. So you have a long string of A's or T's or C's that is known as a homopolymer. All of those bases will be incorporated, and it's very difficult to discern the difference between 10 and 11, 19 and 20. The second weakness was that even though there were dramatic improvements on this machine, This improved from being able to do 20 megabases of sequencing per run to 500 megabases, but there was no room for further improvements in this. Now, as this evolved, and this is shown here, they started as a GS20, a 20 megabase sequencer, but very soon by increasing read lengths, they got this to be a 100 gigabase sequencer. Then, when I say current generation, this was the final generation of that machine. They coated the plates with titanium. The machine became the GS Flex Titanium, which could generate 500 megabases of sequence. And utilizing this machine, the second named person whose genome was sequenced was Jim Watson, and this was done on these genome sequencers at a cost of $1 million. So this is at least a 200-fold improvement, but again, can we bring genome sequencing to be even less than that? Because at $1 million, this is still way too expensive for this to be done, except for perhaps the richest people on the planet. Well, the second sequencing machine that became developed was called the Illumina Genome Analyzer. It was actually developed by a company called Solexa, but Illumina purchased Solexa for $600 million to get access to the technology. The Illumina machine uses bridge amplification, as I showed you before. On the left is actually the individual flow cell that is present in the Illumina machine. There's actually 8 lanes in it, so you have the capability of putting different samples in each lane. This is then placed into the machine, and the Illumina had a number of key advantages over the 454 machine. The first one was that all 4 bases, because they were put with different fluorescent colors, could actually be put in together. You didn't have to do them sequentially. The second thing that was a really important advantage is the fluorescent groups that were added to each of these nucleotides was placed in a position to inhibit more than a single base being added. So in one fell swoop, they made this much more efficient. They also made it so that you could only add 1 base at a time. So they solved the homopolymer problem. What's shown on the right is the very first base is incorporated. After that base is incorporated and you've taken a picture of it, you can add chemicals to remove the fluorescent group. Once you've done that, the base is actually ready to accept the second base, and that base can be added. Again, a fluorescent group is put on a blocker, so it's a base at a time. And on the right, you show the sequence of the first round, the second round, the third round, the fourth round. And if, for example, you take the little dot on the left, the first base incorporates a G, the second is a C, then a T, then a G, then an A. That is the sequence, GC, TGA of that one position. The advances on this machine, because the very first machine was capable of improvement over the 454 machine, it could generate a gigabase of sequence, which was 48 million clusters on the 8-lane flow cell, but the read length was very short. It was only 31 base pairs. But over the next 3 years, they increased the number of clusters and read length to over 100 base pairs, which increased the number of gigabase sequence at higher accuracy. A few years later, a HiSeq 2000 was the second generation Illumina machine. This had a number of dramatic improvements, including running 2 slides, had a much greater sequence output, and as a result, even though the very first machine was only capable of a gigabase of sequence, when the HiSeq 2000 started, it was capable to do 200 gigabases of sequence. However, once they started improving, what eventually you reached a point, and this is shown on the bottom right, the number of clusters was so great that it was very difficult to discern one cluster from another. Illumina took an idea that Rade Dramanik first developed for complete genomics, which is instead of having just random clusters on a slide, to actually amplify in basically a pattern flow cell that has tiny little dimples. What you do instead is you amplify the DNA in the dimples, and when you get the sequence, or when you get the signal coming out of that, and this is shown in the middle, you can see a much more defined feature with ordered spacing, which made it easier to discern one from another and get a much higher number of simultaneous sequences that could be generated. The current generation of the Illumina platform is the Novaseq platform, and this is one of the machines shown here. Actually, there were 2 Novaseq machines, the 5000 and the 6000. The 6000 is the highest throughput machine, And one of the nice features of this is both of them have the capability of running different sized flow cells, depending on how much sequence you want to generate. But with the highest output flow cell, which is shown on the top right, it was capable of 3 terabases of sequencing. And since the machine ran 2 of these, that means the machine can generate 6 terabases of sequence output per run. This is a 6,000-fold improvement over the original sequencing machine from Illumina. What is shown here is the output that was generated first on the Genome Analyzer, which started at a gigabase in about 2007. By the time the machine retired, it had increased its output to about 60 to 80 gigabases. The HiSeq 2000 started at 200, but with improvements was able to break the terabase barrier to generate more than a terabase of sequence. Now with the Novaseq machine, this is capable of generating even 6 times more than that, 6 terabases of sequence output. Another platform that was developed was also developed by Jonathan Rothberg, so it used many of the same capabilities of the sequencing that was done on the 454 machine with some improvements. One of the first was the sequencing is actually done on chips, tiny little chips that look very much like computer chips. They look like them because they're actually made the same way. What you have is a wafer, which instead of being used to make computer chips, is being used to make these tiny little chips that have basically a bunch of little dimples in them. Each dimple is the equivalent of a pH meter. The reason for the name ion torrent is that when a nucleotide is incorporated into a growing chain, a hydrogen ion is released. If you have a tiny little bead, smaller than the beads on the 454, that has a base incorporated, all of the amplified molecules on that bead will release hydrogen ions, and it's the ion torrent which changes the pH in the well. So you put that all together, you basically are incorporating a nucleotide, releasing a hydrogen ion, and that is detected as a change in the pH in that well. You then use the same methodology that 454 utilizes. You flow an A, a T, a G, and a C. Which of those bases gives you a change in pH? The problems was the output on this machine is still a fraction of what Illumina is capable of. They use the same nucleotides that are used in the 454 machine, which are unblocked, which means that if you have a long stretch, a homopolymer, all will be incorporated, and thus it's difficult to determine the precise sequence of long homopolymers. In addition, it's still cumbersome as compared to Illumina because you're still doing the emulsion PCR. And as a result of this, Illumina continues to totally dominate the massively parallel sequencing space. There are less expensive first-generation sequencing platforms because most of the machines I've been describing before are high-output machines that are relatively expensive. A company, Genapsys, is developing a low-cost machine, but has a low output, and that raises the question, how does it stack up against Illumina or Ion Torrent in this space? Illumina has an electronic sequencer that is called the iSeq 100, which is capable of 1.2 gigabases. It does 2 times 150 base pairs of sequence at the end of every fragment in 19 hours. Ion Torrent now has a Genexus system. This is an integrated lower cost system which can supposedly get specimen for a clinical specimen to a report in a single day. It's an automated workflow at a lower cost. So what to sequence? You now have machines that are capable of generating up to 6 terabases of sequence output. You also have some of these lower output machines if you don't need to sequence that much. Well, because you have these machines, you can now tackle a variety of things, everything from a whole genome. And when I'm talking about genomes, it could be the genome of a human, it could be a genome of an animal, or it could be a very small genome like a microbial genome. The only difference is the size of the genomes differ. Perhaps whole genomes is too much information and you don't need it. Well, there are capabilities to sequence defined portions of the genome. And by defined portions, that could be a few genes all the way up to the exome, which is every single exon. There's 200,000 in the human genome. You may, instead of being interested in the DNA, be interested in the RNA, what is actively being transcribed. That is the transcriptome. Now, as I mentioned earlier, there's more than 4 bases. There's also that 5th base, 5-methylcytosine, which is basically the epigenetic modification. That is known as the methylome. And with sequencing, depending on what you want to do, you can choose these different things. But what are you trying to determine? Do you need the sequence of the entire genome to answer your question? Is the structure of that genome important? Because short-read sequencers are very good with sequence accuracy, but since the short-read sequencers are not very good for characterizing our genomes or any complex genome that are highly filled with repetitive sequences. Well, let's talk about WGS, whole genome sequencing. To do this, even though the genome is 6 gigabases in size, the 3 gigabases from your mom and the 3 gigabases from your father, you need to sequence more than that because these are random fragments. So we think of about 100 gigabases of sequence, which is a 30x whole genome sequencing, is enough to generate the sequence of a genome. However, if the genome you're characterizing has heterogeneity, and this is certainly seen in many cancers where you get a group of cells, but they're not all the same, you will have to sequence more. But when you've generated all that sequence, how does one assemble that sequence together? How do you analyze the assembled genome? And again, raises the question, what are you looking for? Well, there are problems with whole genome sequencing. With the pattern flow cells on the Novaseq 6000 machine, the actual sequencing cost has dropped from hundreds of million dollars to only $475. However, that is just the sequencing cost. How much to assemble the genome and interpret it? What happens if you're looking for alterations outside the coding sequence? What is their clinical significance? Only about 1 to 1.5% of the genome actually codes for protein, so the majority of the genome, 98, 99%, is outside of that coding portion. So what does it even mean? And the analysis of that is not trivial. There's also storage of all the data. So as a result of all these things, most of this means that whole genome sequencing, except for specific instances, is not yet ready for primetime clinical practice. Where is it being used? Well, the clinical use that one of the most exciting areas is from the very pioneering work of Stephen Kingsmore and his colleagues, where they have been using this in the neonatal intensive care units. Children are born that are failing to thrive, and people want to know right away what's wrong. Parents are hysterical, and they also want to know. And in this instance, Stephen Kingsmore has developed technologies where he combines the Illumina machine in a fast run mode to generate sequence data, to interpret that data, and have it all done in, this is an older slide, 26 hours, that is now in less than 24 hours. As a result of this, this work became a part and went into the Guinness Book of World Records. However, when you do this type of sequencing in the NICU unit, you traditionally will do the sequence of both parents and of the child. The total cost of that today is about $15,000. As a result of that work, we're starting to realize that this might be the model where neonatal intensive care units start to look at their children who have problems. They're also exploring whole genome sequencing in the context of rare diseases to find out what is wrong. Right now, one of the largest uses of whole genome sequencing are in population-based studies. For example, Genomics England, in collaboration with the National Health Service, is planning in the next year or two to do 500,000 genomes to sequence them. They've actually completed the sequence of 100,000 individuals from the United Kingdom, but they have a collection of 5 million people from the UK and plans to actually sequence all of them. And the idea will be to see if whole genome sequencing can transform clinical practice and helped transform the National Health Service. The United States has a project that is known as All of Us, and this is a United States collection of 1 million people from the US linked to genomic information. And across the world, there are about 50 different projects, and it is estimated by several people that within the next 5 years, this type of work will lead to the sequence of at least 50 million genomes. This is the largest use of whole genome sequencing that is occurring today. As I mentioned earlier, Illumina actually has a monopoly on massively parallel sequencing. They themselves estimate that they have about 90% of the sequence market. I believe it's probably even greater, 95%. And while there are currently first-generation MPS alternatives, Illumina is controlling the sequence space, and up to now was the only viable platform for whole genome sequencing. The Ion Torrent system, and perhaps the Genapsys system, are much more lower throughput machines. They could be used for clinical sequencing of smaller things like small gene panels, but certainly for whole genome sequencing, there is only Illumina. And in spite of the fact that the other machines could be used for smaller projects, Illumina still has a monopoly. This does not serve the scientific or clinical community, because something as important as sequencing, which could become an integral part of healthcare and well-being, should not be controlled by a single company. Well, is there an alternative? And the answer is yes. That answer is the BGI, or the Beijing Genome Institute. They have a variety of million genome initiatives, from looking at 1 million newborns to look at millions of plant and animal genomes, as well as the many millions of microorganisms. Well, the BGI is actually centered at a place that has the China National Gene Bank. And this is a huge storage facility that has 20 million samples stored for sequencing. They have large numbers of sequencing machines, and they have the capability to generate a large amount of data. What BGI did was they purchased the company Complete Genomics that was developed by Rade Dramancic and co-workers, and they then started to utilize the Complete Genomics sequencing platform. That platform is critically dependent on the use of DNA nanoballs. And again, as I mentioned, it's a novel method to amplify fragments using a 5-polymerase to linear amplify templates to 300 copies. The amplified DNA folds into nanoballs, hence the name. It became DNB sequencing, which can then be deposited on pattern flow cells. The original BGI machines, BGI again purchased Complete Genomics, utilize a chemistry that was very similar. It was a sequencing by synthesis that will certainly have patent problems against Illumina. However, very recently, Rade Dramancic and co-workers have developed an alternative method for doing sequencing to try to get around those patent issues. And what they do, this is called cool NGS. Cool NGS, because the actual nucleotide incorporation that occurs doesn't occur with a nucleotide that has any fluorescent on it, any radioactivity. They simply use nucleotides that have a blocking group on them. It's a reversible blocking group, and those bases are first incorporated. After a base is incorporated, a growing chain, because there's a blocking group, it can only add a single nucleotide. What happens after that is they basically use antibodies, which are base-specific antibodies that can then bind to that block base. Each antibody itself, actually as shown here, has multiple fluorescent groups on it, so you get signal amplification. So you basically use the cold incorporation first, you then follow with the antibodies with the different fluorescent groups on them. You get a fluorescent group attached, you take a photograph of that, and when you're finished, you chemically remove both the antibody and the blocking group, that's the blocking group on the cold base, and now you're ready for the next base incorporation. And this is how the BGI sequencing platform is currently working. They also developed a machine. They have various machines with different outputs, but their highest throughput machine is a machine called the T7. The T7 is called a T7 because it's capable of generating 7 terabases of DNA sequence output per run. So it's quite comparable to the Novaseq 6000. Now, what's very exciting about this is that with BGI sequencing, most of which is done in-house by BGI, but on machines similar or just slightly smaller than the T7, they are capable of doing whole genome sequence for a total cost of $600. This is significantly less than what it would cost on the Illumina platform, which includes everything: library prep, sequencing, alignment, and limited interpretation. Recently, BGI on the T7 machine is decreasing that cost to just $400. This generates what is known as 30x whole genome sequencing, approximately 100 gigabases of sequence output. Well, they're setting up plans to make this more high throughput to enable the various million genome projects, with the goal of generating whole genome sequencing at the cost of just $100. To do that, they recently announced at the Advances in Genome Biology and Technology meeting, Rade Dramancic gave a presentation about the platform that is shown here. This is the platform that basically modularizes the DNB-based sequencing, the DNA nanoball sequencing. There's a robotic arm in the middle. The slides, there are multiple slides that can be run, and this thing has tremendous output. This has output that is on the order of 50 to 60 terabases of sequence output. And with this type of machine, they will be able to generate genomes for a cost of just $100. The advantage of the sequencing platform is there's no PCR in the amplification process. As a result, there are less PCR-induced errors when you amplify your individual fragments. As a result of this, with no PCR-induced errors, there are less problems when you put barcodes on different sequences from different individuals and put them all together. You have less barcode misassignment. Cool NGS also uses multiple fluorescent groups on each base-specific antibody. As a result of this, they get signal amplification, which gives them the capability for further improvements. The DNB-seq-TX, the TX for high throughput, can do 30x whole genome sequencing for just $100. Well, the current generation of the DNA nanoballs is 200 nanometers in size, and that gets a sufficient signal. But because the antibodies have multiple floors on it, it should be possible, and Rade Dramancic was talking about this, to make even smaller nanoballs that are just 100 nanometers in size. When they're that size, you'll get an even greater sequence output. So there's the capability of getting genomes for significantly less than even $100. Competition is good. As BGI ramps towards providing a genome for $100 all in, Illumina is going to have no choice but to try to compete or to try to inhibit BGI from selling the machines due to potential patent issues, which are questionable. We have seen a reduction in whole genome sequencing from $3 billion to $600 or less in just 19 years, with the capability of the $100 genome being very close. Well, once we have the $100 genome, how long until it's even improved further? We've gone from million-fold improvement in sequence output from just 20 years ago, and we're almost at the dawn of the $100 genome, and with some of the advances that Rade Dramancic is talking about, we could see a sub-$100 genome. Why is the $100 genome the turning point? More than anything else. For many years, the target seemed to be the $1,000 genome. Well, the first reason that it's a true turning point is when genomes just cost $100, it really facilitates population-based genome sequencing studies much more rapidly. The plan to sequence 5 million individuals in the Genome England project will cost considerable amount of money when it costs $1,000 to $200,000 per individual. But if it costs just $100, the possibility is that it's going to be easier to do, less expensive, and many other countries will do similar efforts. And studies like the one from Genomics England and the National Health Service will reveal the value of whole genome information to direct health care. And at $100 or less, whole genome sequencing could become a routine part of our medical record, and this will really get us much more towards a fully envisioned personalized medicine. More importantly, we can start to transform the paradigm of healthcare, of one where people are treated when they have disease, instead to having a better understanding of who might develop disease based on genome sequence. And we may also have the capability of basically having healthier populations based upon information from genome sequence, and possibly other information like what would be ideal diets, what would be ideal exercise regimens, which might be very much dependent on the genome that you've actually inherited. Well, what aren't we seeing with all of these first-generation massively parallel sequencing technologies? The two major competitors in this area really are Illumina and BGI. They only sequence fragments that are relatively short, and taking these short fragments and looking at genomes that is 45% repetitive is very, very difficult. All the first-generation MPS, certainly Illumina and BGI, are very good for base calling accuracy, but they're very poor for characterizing large structural alterations, which are an important part of cancer and many other disease states. There's other problems with the first-generation MPS platforms. All are short-read technologies, so again, it's difficult to generate the complete genome sequence of a complex genome or of a highly rearranged cancer genome. In addition, you cannot, when you use this for RNA sequencing and looking at transcripts, many of them are considerably larger than the sequence length of the first generation technologies. So when you sequence, if you get a tiny little piece, you don't know which transcript isoform it came from. So if you could sequence longer pieces, you could determine exactly which isoform you're looking at. The other thing is that it's not possible to directly use first-generation technologies to look at genome-wide methylation. With these technologies, you have to take the genome, sequence it, take that genome, bisulfite, modify it to change all the 5-methylcytosines to uracil, and resequence it again, so you actually have to sequence the genome twice. Well, this leads rise to second-generation MPS technologies, whereas all first-generation technologies, which are highly dependent on amplifying DNA molecules, second-generation technologies are single-molecule sequencing technologies. Because you're doing single molecules, you don't have to amplify them, which means that you can look at much larger pieces, and because you're not sequencing copies of copies, you can actually sequence much greater lengths. And actually, both of the technologies that I'm not going to describe, but I'm just going to talk about briefly, can now sequence fragments that are sometimes in excess of 1 million base pairs in length. The two main platforms in the second-generation MPS technologies are Pacific Biosciences, and we're very pleased that Dr. Jonas Korlach, my friend who's the Chief Scientific Officer of PacBio, is going to have a talk in this webinar where he's going to be talking about some of the exciting things that PacBio is doing in the sequencing space. The second sequencing company is Oxford Nanopore, and my colleague, Dr. Mark Ebert, who's at the Mayo Clinic, has been using Oxford Nanopore sequencing to reveal portions of the genome that are completely hidden to short-read technologies. The last two parts of the webinar that are part of what I'm discussing are the clinical uses of massively parallel sequencing. And my colleague, Dr. Matt Ferber, who's at the Mayo Clinic, will talk about his work to develop analytical and visual tools for interpreting massively parallel sequence data. My other colleague, Kevin Holling, also at the Mayo Clinic, will talk about his work because he's developed RNA-seq, RNA sequencing, as a clinical assay to detect translocations in cancer. I thank you for your time and your attention.
Thank you, Dr. Smith, for that outstanding presentation. For additional informative presentations, view our agenda on our events page. As a final reminder, our speaker will follow up with any questions you've submitted via email. Thank you again for your participation.